All right, hi there, AP Bio. This is your DNA replication lecture. It's been a long time since I actually did this in the classroom because last year this was a snow day when we were learning about this material. So there's an old video out there in my YouTube channel of this as well, but I thought, why not do it again? So I've been teaching in a while, so it might be nice to talk to students. This can be found on Schoology, all filled out. I've made a few minor little changes here and there, but the list of what these enzymes are and what they do and the colors, you can always do your own colors, but this isn't changed much. I thought I would start down here at the bottom and talk to you about the structure of DNA again. We've been talking about this a lot this year, so hopefully you're like, okay, I know, Miss Nev, I got it. 3 parts to a nucleotide, right? Sugar, which I've colored yellow, phosphate out here, and then the base is attached to the other side of the sugar. So the sugar's kind of the anchor. The bases for DNA, A, T, C, and G, and now they've got more specific names. The ones that the nitrogenous base has 2 rings in it, so not the ring that is the sugar, but the ring that has the nitrogens in it, that's the adenine and the guanine. Those are the purines, and the thymine and the cytosine are the pyrimidines. So you can fill that in right here. Purines A and G, pyrimidines T and C. And a way to remember that is a couple of little tricks. Pure as gold, so pure like purines, A and G as gold. But that doesn't tell you that they're the double ringed ones. But PUAG2 works. I think gold, you always want to have more of it, right? So the ones with 2 rings are the gold ones. But that can be a way to help. So when bonding a double-stranded helix, we know the complementary bonding is A to T and C to G, but why does it work that way? Well, it's because they end up having this nice equidistance across from each other with a double-ringed purine and a single-ringed pyrimidine every time. If you try to put the 2 purines together, the A and the G, you end up with a helix that's too wide. And if you try to put the pyrimidines together, you end up with a helix that's too narrow. Oh, low power mode on my phone. Stand by. Charge cable not far. There we go. All right. So that lumpy bumpiness is actually what enzymes that look to do DNA repair are looking for. If it's equidistant across side to side, that's good. We've matched the right ones together. Never you mind if we change one of them and replace the wrong one, that's where mutations come in. But keeping the right distance of the helix of that parallel twisted ladder is accomplished by a purine bonding to a pyrimidine. And they bind with hydrogen bonds. A to T has 2 hydrogen bonds, and C to G has 3 hydrogen bonds. The other structural thing to notice here is the directionality. So you can see there's numbers. First off, cross off the O, that's for RNA, ribose sugar. We are talking about DNA, so this is deoxyribose, cross that oxygen off. And what I want you to notice is these numbers, 1, 2, 3, 4, and 5. And that's numbering the carbons. So there's a carbon right there, and a carbon there, there, and there. 1, 2, 3, 4, 5 carbons here on this ribose sugar. And we number them in that order. And so the base is attached to the 1' carbon, but the backbone is going to be formed this way. Okay, top to bottom, and going from 5' red down towards 3'. So I'm going to keep repeating that again and again, where when we are looking to attach a new nucleotide to an existing strand of DNA, we're going to be attaching at the 3' end. We build 5' towards 3'. So now we will attach a new 5', stick out a 3'. And so you can see that over here. I know that they all look right side up, but look at the oxygens. If the oxygen in the sugar is the top, like we're going to build like down towards its feet, right? So like the 5' is the carbon that's connected to the phosphate. That's up towards the top by the oxygen here. And then the 4 isn't attached to anything, it's just in the ring. And the 3 is going to have an oxygen sticking out that will attach to the next phosphate. So there's that 3' oxygen coming down. Phosphate is connected to the 5', and we're sticking out 3' again, waiting. So DNA runs a lot like a two-way street that has directionality to it. They're not parallel going the same way, but they're going in opposite directions. So as we build new pieces, we're going to have to build in that order. So what happens initially? Our DNA is linear, so you can imagine a bunch of DNA, chromosomes like this. And again, this is DNA replication happening in S phase, which is in the middle of interphase. So in our cell cycle, we have G1, S replicating DNA, G2, and then mitosis, and we would like go around again. So our DNA isn't yet totally condensed, but we're going to like open up and pull apart those hydrogen bonds. We're going to do it there, we might do it here and pull those apart. And you can imagine it like a rope. Oh my gosh. Hey, speaking of yarn, right here. If you're trying to open up and get into the middle, you're going to have like a lot of tension upstream and downstream of that opening up section. We call this the fork right here. Oh gosh, this is terrible because I only have one hand. So the replication fork, I've opened this bubble, and this would be a replication fork, and this would be a replication fork. Not here in the classroom with my original model of this, and I don't want to ruin my yarn for my nice sweater. I'll show you how my sleeve here at the end. But you're gonna bump into each other as you meet across, and you'll come across the ends. If you were a bacteria that has circular DNA, you would just start in one position and go that way and go that way, and then until you have two separate loops. So, all right, so it's our enzymes involved. Single-stranded binding proteins. I've got those here, and also here and here. They're basically helping to stabilize the strands. You could see when I pulled apart the yarn, let me get one more actually. No shortage of yarn here at my house. When you unwind and you're trying to get to the middle, you're going to have to be stabilizing these other ends. So those single-stranded binding proteins are kind of breaking the backbone and maybe untwisting some of the tension and just sort of holding it in place so that other enzymes can do their job. Helicase here, shaped like an H, I've got one here on the top strand going to the right, and one on the bottom strand going to the left. That is going to be breaking breaking the hydrogen bonds, and it's going to be opening up the DNA helix. You've probably heard cheesy jokes of like, oh, I want to get into your genes, and I'm helicase, and I'll unzip them, and blah, blah, blah. So there is more tension that gets accumulated, and that's what topoisomerase is for. So I've got a big one up here, and a big one down there, kind of moving outwards as the loop gets bigger and bigger. And this is, again, helping to relieve the strain and the tension. It's basically, again, cutting the DNA helix, letting it untwist, and then sealing it back together so that we can continue to unwind internally. And it relieves the tension farther upstream. So, now that we've opened it up, I want to draw your attention to the 5' to 3' directionality on the diagram. So this top strand out here, I've colored it dark blue. I put a 5' on the top, and then that means a 3' pointing this way. Connected to a 5, connected to a 3, 5 to 3. So the sticking out end over here would be 3. And because of the directionality of DNA, that means the bottom side here is going to be 5' going this way to 3', okay? The outside I've colored blue, and we're calling this the template strand. When DNA gets replicated, it's a semi-conservative process. So the template strand will exist, it will separate, and then we'll bring in like 2 new sets of base pairs to then wind up, and now you have half old and half new on each new molecule of DNA you've made. We call this semi-conservatives because we've kept some of what we had originally, but we have new stuff as well. So I've got that over here. The inside ones I've colored teal. I made a poor choice probably. I don't know if you could see it on color. This is blue and these are teal. And the inside ones are teal where they're smooth. The stripy ones are going to be something else, but the inner ones are leading and then lagging strand that I've added in here as well. So the template strand will be copied down in a 5' to 3' direction. So if I've got 5 up here and 3 over here, well, to copy 5' to 3', I've got to start to the right and go to the left on the top strand, which is exactly what happens. What's going to come in and get it started is primase. So primase with a P here, it's the enzyme that lays down the first few nucleotides to get the other enzyme something to grab onto, okay? And they're not laying down DNA, they're actually laying down RNA, which is why I have them stripy and a different color, okay? So they're actually RNA bases that are complementary to the DNA right here, just a couple, 5 or 10, because DNA polymerase 3 is kind of a dum-dum of an enzyme. You'll love the Amoeba Sister videos of this. It's just going to look, where do I go? I need something to hold on to. And that's his attitude, basically. So primase lays a few down, and then it would leave. Okay, this is static drawing, so you have to imagine it being gone. And then DNA polymerase 3, shaped like a D here, will lay down the new nucleotides of DNA in the 5' towards 3' direction. So right here, if that's 3 and we're going the other way, this was a 5' base heading this way towards a 3'. So we can now attach the phosphate of the 5' end here and point downwards towards 3' over here. This is going to be an open road. This is what we call the leading strand. So as the replication fork expands and moves farther to the right, this is what I mean by an open road. It's just going to lay down more and more track and keep it going. There's nothing in its way as the fork opens up. But we started here, and we're also opening up to the right as well. There's all this stuff behind that it didn't take care of if we started the primase here. So what these are, this is the lagging strand, okay? Leading strand has no problem. Leading strand built in a continuous piece as the replication fork extends. This I would call the open road, okay? The lagging strand is built in little chunks that are trying to catch up, okay? And as it primase lays down, it's going to be able to build some until it bumps into the other primer. It's, oh, I'm done. That's all you needed. But then more road opened up behind it. Okay, lay down a primer farther back and then build with DNA polymerase some more DNA. Oh, that's all I need. So a lot of this is real kind of herky-jerky and start and stop feely, but it's filling in the gap that is getting created as this helicase opens up to this direction, because you still have to be building 5' to 3'. You're opening up new 5' opportunities as you go, but you have to continue to build back what you left here. So the lagging strand built also in 5' to 3', but in small chunks until it butts up to already completed parts. So I like to think of the lagging strands as the cleaning crew. And this word in the middle here is my favorite. All of these little chunks of fragments are going to get all put back together in a second here, but we call these fragments the Okazaki fragments. It was, I believe, a husband and wife crew. Don't quote me on that, but I love that name, and I love that, I don't know, it's just, I think it's a great statement. I think it's cool. So we're expanding, we're building leading strand this way to the left. We can do that also to the right on the bottom strand here. So we can extend these permanently in these two directions. Let me make my fingers go like, woo, that way. But we're gonna have to play like catch up and clean up behind them, okay? we're also going to have all of these breaks in the DNA. So that's what ligase is for. Ligase to ligate, to glue, not to lyse, which is to break, but lig, like gluing. This is going to seal the gaps formed in the backbone because this RNA and DNA like aren't bound together, and we're going to need to bond these like final like sugar to phosphate bonds along the backbone. And not pictured in the diagram because it's already a little crowded is DNA polymerase 1, so it has a little Roman numeral 1. It's going to be the one who swaps out the RNA nucleotides for DNA nucleotides. And nuclease, which is helping any repair or damage. What nuclease is looking for is these bumps and like indents in the backbone. It's looking for uniformity in the backbone. If we have a purine and a pyrimidine bound together each time, we're good. And so nuclease can kind of like roll along the completed helix and look that it's the parallel distance apart. So that's DNA replication. Pretty quick, simple lesson. I don't think I need to go over like A to G, no, I'm wrong. A to T, C to G. You could practice that till the cows come home. I think we might be able to create some kind of a fun, like, discussion, like, lesson with creating protein messages with each other. So kind of like decoding and sending fun things that we're doing. So I hope you're staying healthy and staying home. I'm going to blow my nose now, and we'll upload this video. It's 4:40, should be up there around 5 o'clock. All right, so fill this out. Read the chapter if you like. I don't know what chapter it is. I'll check in the book. All right, take care, guys. Bye-bye.